Everyone. Right, so it's 8:10 and let's begin. So welcome to the first SG STEM session for 2021. I think a couple of weeks back, Kanan asked, hey, want to do SG STEM again? I was like, oh no, I'm in the middle of work and all that because phase 3 has started. And so Kanan is really driving most of this. So thanks to Kanan. Where's Kanan? Say hello.
Hello, everyone. Hope everyone's doing fine and chipper in the new year.
Super. Very lucky to have with us Dr. Darren Yeo, who is based in the NUS Evo Lab, who will be telling us more about uncovering Singapore's biodiversity through environmental forensics. So Darren has been working in biodiversity for a long time and has great interest in biodiversity. And I first knew him when he was in, I think, secondary school, when he was volunteering with the museum for Project Semakau. And
oh wow, that's
yeah.
a long time ago.
And since then, it's been our regular volunteer and has really grown into a scientist now. So he'll be telling us more about his work, which we are excited to hear. So welcome, Darren.
Thank you. Oh, should I?
Yeah, you can just share over here.
Okay.
Yeah, I'll stop sharing mine.
Okay. All right, can you guys see the slides?
Yep.
Okay. All right. Firstly, thank you everyone for joining me today, and thank you Kanan and Marcus for inviting me. It's really great to be here. I'm Darren. I'm from NUS Evolutionary Biology Lab, working under Professor Rudolf Meyer. And today I'll be talking about uncovering Singapore's biodiversity through environmental forensics. So I have to quote this because there's actually a term called environmental forensics. And I'm just using this as an analogy. Yeah, so just a bit about myself. I started my undergraduate work with the NUS Evolutionary Biology Lab, working firstly on ordinates, which are dragonflies and damselflies. So these are like my favorite insects. And then after that, I joined PhD at NUS and Imperial College, working at the London NHM. So my PhD work there was mainly on the species discovery of diverse insect groups, such as flies and beetles. So hold on. Oops. Oh yeah. Okay, so before going into the main topic of today's talk, I'd like to talk first a bit about how people commonly view biodiversity. And if you do a Google image search of biodiversity, these are some of the first images you will get. And you can see that most of these images are dominated by charismatic vertebrates, your pandas, your lions, your elephants, and you don't often see many invertebrates. And also when we think about biodiversity, we tend to think of zoos, museums, or perhaps nature reserves. And in other words, to a layperson, biodiversity tends to be peripheral to our daily lives. And this is, in Singapore, this is reflected in our land use allocation, with nature reserves only taking up about 4.5% of our total land. However, I think we need to rethink our idea of how we view biodiversity. So for one, we actually know very little about the species around us. Most of the species out there are still new to science, and we actually don't even have a good estimate of how many species there are on Earth. Out of what we know, most of species are actually invertebrates, mostly dominated by insects, and the vertebrates or chordates actually form a very small proportion. And even in terms of biomass, most of it is actually dominated by plants. So in other words, if we are focusing mainly on the charismatic vertebrates, we are actually having quite a narrow view of the world's total biodiversity. So furthermore, biodiversity is not something optional or nice to have. It's the World Economic Forum actually ranks biodiversity loss as one of the top 10 risks over the next 10 years. And the same report ranks biodiversity loss as the most impactful and the third most likely risk for the next decade, owing to implications for the collapse of food and health systems, as well as disruptions to supply chains. So the two main points so far is that biodiversity consists mainly of small species-rich organisms, most of which are still very poorly known. And that biodiversity is actually crucial to our existence and something we cannot afford to lose. So in order to better preserve and rehabilitate our ecosystems, we need to actually understand them better. So knowledge is power in this case. In the context of Singapore, we have actually lost most of our natural spaces, and therefore we need this knowledge of our ecosystems to sustainably manage what we have left and prevent ecosystem collapse. However, ecosystems can be extremely complex. So like a messy crime scene, there's often more to it than meets the eye. So thankfully, there are modern DNA techniques that can provide clues as to what is really going on. So if you think about the context of the crime scene and let's say you have a sample, like a hair sample or blood stain, people tend to think that we start with collecting DNA from that. But many forget that you actually need a DNA database to compare this sample against to make sense of it. So much like DNA work with biodiversity, we actually need to build a database first in order to enable a lot of the DNA work that comes later. So in order to build this database, we ideally have a specimen, and ideally that is identified and properly described. Then we just need to sequence a DNA barcode for the database. So what's a DNA barcode? It's basically a short gene fragment with enough genetic variability to distinguish species. So you can think of it as like a supermarket barcode that corresponds to an item on the shelf. However, we have to remember that most of biodiversity consists of species that are still small. They are small and still not yet known to science. This means we neither have a species description nor a DNA barcode. And one reason for this is that the traditional workflow for sorting species is usually very time consuming. So if you talk about like an insect sample, if you collect this from an insect trap, a malaise trap for instance, you see that it's a ton of specimens, it's extremely messy and chaotic. And typically you get a taxonomist who has years of experience in this group to look at these specimens and group them to morphospecies based on morphology. So what is morphology? It's basically the shape or structure of the specimen and basically just how it looks like. So many of these specimens can be very, very similar, even though they belong to different species, and this complicates things. So once we have these morphospecies, usually we can then sequence a DNA barcode. And this barcode is often used to verify the accuracy of the morphospecies. So this is a very slow and expensive process because there are very few expert taxonomists in some of these groups, and the time is very costly. However, we can speed up this process with next generation sequencing of DNA barcodes or NGS barcodes. So instead of just sorting things using morphology, we can sequence barcodes for every single specimen. This is possible because NGS makes everything much cheaper. Then we can use similarities and differences in the DNA barcodes to group specimens into putative species or what we call here MOTUs. And this makes it much easier for taxonomists to verify. And yeah, basically it makes their lives a lot easier. Ultimately, what we're doing is to reduce time and cost so that studying small and species-rich groups becomes much easier. So a lab has done this for insects and other arthropods in Singapore. We've sampled on the island for more than 8 years or so. We've collected more than 150,000 specimens representing more than 10,000 species. And these have been from multiple different habitat types in Singapore. So the species we found most of them have been imaged and are available to the public at the Biodiversity of Singapore website, pioneered by Dr. Ang Yu. And yeah, if you guys have the time, just do pop over there and take a look. So now that we have these species units, we can also explore broader trends, and we've done this particularly in Singapore's mangroves. So typically, people don't usually associate mangroves to high insect species diversity because mangroves have high salinity and low plant diversity. However, what we found was that the mangroves actually have a higher species richness than expected. If you look at this graph here and look at the green line, it's roughly 2/3 of most species-rich forests. But what is actually more interesting to me is that the mangrove insects are extremely unique. So if you look at this plot, this is an NMDS plot, which represents how the insect communities are different or similar in 3D space. So if you just focus on the points in green, you can see that they're very separate or very distinct from the other habitat types. So this means that if tomorrow we suddenly lost all of our mangroves, we would have lost a huge proportion of our insect diversity because they are found nowhere else. And another thing we can do with these DNA barcodes is we can match different life history stages. So what does this mean? So going back to one of these very pretty damselflies, you might not expect it, but the limb looks very different. And often we have a very good idea of how the adult dragonflies and damselflies look like in Singapore. But we know very little about the nymphs because they are much less visible. Fortunately, both the adults and the young will have the same DNA. So if you sequence the DNA of the adult and then you sequence the young, and if the DNA is the same, then you know that you have a match there. So using this approach, we've also managed to match many more dragonfly and damselfly species in Singapore. And this now allows us to identify the nymphs species. So if you're curious, these nymph images are also on the Biodiversity of Singapore website. Right, so once you have a good DNA database, what you can do is that you can collect a biological sample from the crime scene, such as a blood stain. You can sequence the DNA from the sample and match it to the database to identify the perpetrators. So similarly, in nature, the DNA is everywhere and in all biological samples. So for instance, even soil and water would contain DNA of organisms that have passed through them. So this DNA is commonly referred to as environmental DNA or eDNA. So one of fellow postdocs in the lab is investigating the eDNA in Singapore's freshwater bodies. And just by taking a water sample and sequencing the DNA in it and comparing the DNA to a reference database, you can actually find out what's in the water body. And this has important implications for detecting potentially invasive or nuisance species, or just a broad survey of what is usually in a baseline survey of what's in the water body. And of course, another source of DNA is poop. So another postdoc in the lab, Amrita, has been studying the Raffles-banded langur, which is one of Singapore's most iconic species, just by collecting its poop. And from the DNA in the poop, she's managed to obtain information about what it's feeding on, as well as the gut parasites, which also gives an indication of the host health. So you can get quite a lot of information about the host just by studying the species. So the poop will also contain DNA from whatever made the poop, right? So using the host DNA, postdocs Andy and Amrita have also found that 3 subspecies of langur found in the region are genetically distinct enough to be elevated to species. And this has important implications for conservation. So in conclusion, with the proper application of DNA tools, we can actually better understand what's happening in our ecosystems, from deriving community patterns to obtaining species interaction information. However, I must emphasize this, that building the database is key to tapping into this potential. And currently where we're at is not, it's nowhere close to a comprehensive database for Singapore's biodiversity. And this is where our efforts need to go in order to better utilize the promises of environmental DNA. So in short, we need to redirect our efforts into filling the knowledge gaps in our biodiversity, mainly in the small and species-rich groups, which are still not well studied. And this has to be done soon to avert disaster and ecosystem collapse. So I've come to the end of my talk. Thank you all for listening. And I'll stop sharing that. Yeah.
Excellent. Thank you very much, Darren. That was a very informative talk, and I enjoyed all of the pictures, especially the last one with all of the insects as well. That's great. So let's see questions. If people have questions, please throw them down in the chat. And while people are typing questions, I have a question. So you said that we can use environmental DNA to better, what do you call it, assess and conserve habitats and stuff like that, right?
Yeah.
Are there places in the world where this has worked, or this is something they do as a part of their, what do you call it, conservation efforts?
Yeah, I think so. I think it's been used for a variety of it has a variety of applications actually. So one potential use is actually a baseline monitoring tool. So for instance, if you know that if you suspect there's a risk of a particular invasive species that's coming into an environment, you can start doing these baseline monitoring using environmental DNA to detect when this organism first arrives. It tends to be more effective for a broad study in more temperate regions because they tend to have a more comprehensive idea of the total biodiversity there. So the tropics tends to be more poorly studied, and like I said, you need to have a good DNA database to be able to make sense of what you get.
Okay.
Yeah.
I'm surprised that the tropics are poorly studied despite their species diversity, especially with insects. Yeah, since you said that we have to focus on insects, I'm quite surprised. We have a question from Lydia. Lydia asks, and also there's also Henry's question, who will fund scientists to build the database?
Oh, good question.
Any question about funding is a good question.
I hope lots of people will, but I think this ties into the idea about rethinking our ideas about biodiversity, about something that is actually quite crucial, and that is something that needs immediate attention to prevent our daily lives actually from being affected. So it's a matter of convincing stakeholders and policymakers, perhaps, that biodiversity is not just trying to conserve a rare species, just like a single rare species, but protecting entire ecosystems and better understanding them as well. Yeah. So I think actually last year has been quite a wake-up call with regards to the pandemic and zoonotic diseases, and that we actually need more efforts into, we actually need to study or at least monitor the ecosystems we come into contact with better. Yeah. And hopefully this translates into into more funding.
Definitely. All right, I've got one more question on my side. It's from Audrey. Is the concentration of eDNA sufficient to identify this species?
Yeah, most of the time it is, but it really depends on where you get the DNA from. So for instance, we know that acidic water tends to degrade DNA faster. And especially in warmer climates, DNA tends to degrade faster as well. So you can find DNA there. The DNA there is there all the time, but the time frame in which you are detecting species will actually shift depending on the quality of the DNA you get.
Okay, fair enough. So what happens when you get a mess of DNA? Like how easy or how like complicated it is to like pull out species from it?
I think that one really depends on the database. So you can,
Okay.
if you have an incomplete database, for instance, you can identify things to a higher taxonomy level. So I can, for instance, I can say that maybe I get a Diptera or a fly instead of Drosophila melanogaster.
Okay,
So
so
all
you
I
can.
know is that there is a fly in my sample, but I don't know which species it is.
Okay, fair enough. All right, that's excellent. I think I'll pass it over to Marcus, see whether he has got any questions.
Oh, we've got two more from the floor, which I think we'll take those instead. So one question by Fang Ting is, can you see evolution patterns from the current DNA database?
Oh yeah, you can. So you can actually, so the species in the DNA database, you can potentially study their evolutionary histories by doing more sequencing. You can put them on a tree, phylogenetic tree, and that will tell you how certain things evolve, how certain groups evolve. And this is, I think in our case, it might be particularly interesting in the case of mangroves. So we know that mangroves have quite a particular set of challenges for the organisms that live in there. In them, they need to adapt to different salinity levels, different temperature levels, and this promotes particular evolutionary niche, and this might contribute to the unique communities there. So if you can actually link the how species have evolved to deal with mangroves to and put this and reflect this on a tree, that would be quite interesting, I think. Yeah.
All right.
Hopefully that answers
Okay.
your question. Yeah.
Well, let's do one more, and then after that we'll jump to the trivia session so people don't leave. This is from Philemon. In the local education scene, like secondary schools, biodiversity has been de-emphasized in the curriculum. The administrators had not apparently been convinced of the importance. Any chances there's outreach programs, opportunities for collaboration between schools and the researchers? Very nice question.
Yeah, that's a good question. I think so one way of the way of broadening the outreach for our lab has been to make the DNA process much cheaper and more available to other labs. But another way of promoting this is to engage more citizen scientists. And there is definitely room for collaboration by, for instance, doing bioblitzes. If we have everyday citizens just setting up like a monitoring trap in their backyard or something like that. And then even participating in the sampling process. I think this will lead to a much faster way of addressing these knowledge gaps in our biodiversity.
Okay, fair enough. Thank you, Darren. And I agree as well. I feel like diversity is quite lacking in schools, especially local biodiversity is heavily lacking. It'll be great if we can like push that back in the curriculum. So yeah, that concludes questions on my side, and I think that's it for Marcus as well.
Yep. So thank you very much, Darren, for sharing with us your work and the talk. Maybe like what we do every time after a speaker speaks is that we welcome everyone to say thank you. Then unmute yourself, say thanks to Darren. It's a refreshing thing doing Zoom meetings. So thank you, Darren.
Oh, thank you. Thanks everyone for listening.
Thank you, Darren.
Thank you, Darren.
Thank you.
Thank you, Darren.
Thank you, Darren.
Thank you.
Okay, so I'll request that you mute yourself again because what's happening next is the trivia session, so don't go away. But before we start the trivia, I'd like to issue an apology to everyone again because we've been a bit rusty after 5 months. So if you just join us because of snafu in the links that was sent out, sorry about that. We tried to send out the correct link at the end, and also sorry for the spam. We'll make sure the next one, it wouldn't happen for the next one. So what's happening next? But don't worry, if you missed Darren's talk, it's recorded. It would be posted on the SG STEM YouTube channel, so you can catch up with it once we have processed the video, usually at the end of the week. So next on is our trivia session. So
yes, it is trivia time.
Yeah.
Let me just share screen very quickly. All right, here we are. So sure, presentation view will be so much better. For those of you who know, and for those of you who do not know, we do optional trivia pod contributions, the minimum of $1. You guys can PayNow me or you can PayPal me. Just put your name there as well. I mean, the email will give me your name, so it's fine. So what we do is when you sign up for a trivia session, there, tinyurl.com/sgstem-trivia, you can go and put your team name and your preferred beneficiary. We encourage you to put local beneficiaries because why not? And at the end of the day, all the money in the trivia pot will go to the beneficiary that was picked by the trivia winner. So there have been instances where we have had 2 trivia winners and we just split the pot equally. So yeah, if anyone has got any questions, do drop them below because like Marcus said, it has been a while and I definitely forgot some very important detail, which will bite me later.
So another thing I would like to mention is that there is an extension of the dollar matching grants for local charities. That's why we encourage that. So for every dollar that's raised or donated or contributed to a Singapore-based charity, the Singapore government would match $1 until March 31st, 2021. Right? So there is a change in formats in a way. Great, you asked. So how does this happen? So after you guys, I see that most of you guys have filled up the trivia teams as well as your beneficiary on the trivia page, which I just posted on the link there. What happens is that we're going to play 4 rounds, including 1 bonus round. And each round, instead of having 5 questions, we now have 3 questions. So it's a bit shorter. And today's team is DNA. So after what happens is that there would be you mark your own answers at the end. We're going to review that to you. So it's all an honor code. You take a photo of whatever you've written down as your answer on a piece of paper, or you could text your answers to us at the end. This is just for auditing purposes that we check that we can verify that. And after that, we will share the answers with you guys. Then you self-mark. Everyone would update the Google Sheet on your scores, and then we can declare a winner there. Right, so that is what's going to happen. If you are still not very sure what's happening, just play along. We'll sort it out in the end. It always happens. So why not we just go ahead and see how we go for this.
Yeah, just drop your questions in the chat box and Marcus or I will answer them. And let's go. First one is Darren. Don't worry, this is not a personal question about Darren's likes and dislikes. These are questions given to us by Darren, because Marcus and I got tired after a while of thinking. So question 1, what long-term risk does the World Economic Forum rate as the 2nd most impactful and 3rd most likely? A water crisis, okay, several water crises, cyber attacks, biodiversity, human-made environmental disasters. I want to say it's supposed to be biodiversity loss. I did a boo-boo. So yeah, water crisis, cyber attacks, biodiversity loss, and human-made environmental disasters.
So write down your answers or type it out on your phones so that you can email them to us later. We'll put the email address.
Yep, and don't be like private messaging me for answers. That's illegal.
Yep, and no cheating. So you can't Google the answers or open the books. You could ask your friends if you're in the same team, right? So that is the limits that we have imposed.
Yeah. All right, moving on. If we are too fast, just put in the chat and we'll move back, right? So yeah. Second question, what do we call a short DNA fragment that is variable enough to distinguish or identify species? What do we call a short DNA fragment that is variable enough to distinguish or identify species? For open-ended questions, if you guys want to like contest your answers later with remark, go ahead. Because since Darren will be hanging around, he can tell us whether or not to give you the marks. So you guys can go and be angry with him if he says no. All right, and moving on to question 3. What biological samples can we get DNA from for eDNA work? What biological samples can we get DNA from eDNA work for eDNA work? And the reason Edna is there is because I tried to Google pictures of eDNA, and if you do not put a hyphen, Edna turns up. You have to put E-DNA. But what can you say, darling? No capes.
She can be the mascot for eDNA, I suppose.
Yeah, she totally should be. So A, water, B, soil, C, both of the above, D, others. So you guys can do a fill in the blanks and we will discuss them later. I was quite shocked. I was like, why is she turning up? And then realized that the name was Edna. So yeah. And yeah, so we are keeping it to 3 questions to save some time. So yeah. And second one is net zero. I'm just going to quickly say, does anyone need any questions repeating? No? Okay, let's go. True or false? Singapore made a pledge to a net zero emissions target. True or false? Singapore made a pledge to a net zero emissions target. And this net zero emissions target was is a movement or a plan by the World Green Building Council, which is why their logo is there. Marcus, can you take the question?
Audrey has asked, pledge. What does it mean to a pledge? So a pledge is a commitment, say to a global agreement like the Paris Agreement. So did they make a pledge, a commitment, so to speak, to a net zero emission target?
Does that answer your question, Audrey? New Zealand turned its net zero by 2050 vows into law in November 2019, but with a catch. What is it? A, it will leave out methane. B, they will resume emission levels after 2050. C, New Zealand wants to be rewarded for all the good work it does for the environment. And D, the sheep will have to go. They will have to cull all of their billions of sheep to meet this net zero by 2050.
A sheep that looks pretty cute.
Actually, I like to think the sheep looks shocked at this option. Like they're gonna kill us. It's like the one of the koala, you look the shocked koala, but sheep.
Yeah, hope they don't get cowed.
Yeah. So anyway, it's not like bye-bye, it's like bye-bye. So here, it could be the answer, it could not be the answer, who knows. 3, how many percent of our buildings gross floor area, gross, not gross, floor area will be getting greened to meet our net zero pledge? How many percent of our buildings gross floor area will be getting greened to meet our net zero pledge? When I say our buildings, I mean like buildings in Singapore, not globally. And I assume this is by the pledge, yeah. Okay, moving on. And the last one for A is apps. And first one, which local app came under scrutiny in the last week regarding privacy concerns? Which local app came under scrutiny in the last week regarding privacy concerns? If anything, it should be all of them. You talk about something and wish should be under scrutiny. You talk about something and wish tells you that you should buy it. Why?
Is it WhatsApp? I got a notification from WhatsApp this morning. So what is it? Or was it something that was debated?
And question 2, which video sharing social networking app was banned in India alongside 58 other apps, including WeChat, Clash of Kings, and Mobile Legends? Which video sharing social networking app that's such a mouthful. Video sharing social networking app was banned in India alongside 58 other apps, including stuff like WeChat, Clash of Kings, and Mobile Legends. All right, moving on. The 36th Singapore Bird Race was held on the 5th and 6th of December 2020 with the aim of promoting appreciation, awareness, and conservation of wild birds in Singapore. What app or online database was used to report bird observations? What app or online database was used to report bird observations in the 36th Singapore Bird Race that was held in December last year?
For those for the uninitiated, the bird race is actually an event that's organized at my Nature Society of Singapore, where all participants will go out during a set amount of time. For this is 24 hours, where they would try to spot as many birds as they can. And they record this, and the winner would be the group that has seen as the most number of birds. Right? So there are various categories. And for this year, it was a socially distanced one. And they had to use an online database or an app to report their bird sightings. All right, so which is the app or online database they used?
Right. And it's time for answers. So we will give you guys a moment to discuss or to ask us to show you the questions again if you need any of them for a eyeballing. And then we shall move on to answers. And following the answers, you guys will email us your answers so that Marcus and I may look through them and just double confirm who wins at the end. Yeah. Since there are no strong emotions, we will carry on. For Darren's questions, again, I left out the word loss, but biodiversity loss is a long-term risk that the World Economic Forum rated as the second most impactful and third most likely. And a short DNA fragment that is variable enough to distinguish identify species is known as DNA barcode. DNA barcode. If you guys have contests, you can drop it in the chat. Okay. The third one is water and soil both can be used to get DNA for eDNA work. Darren Sinway wants to know whether they can use SNP for the previous one.
That's short nucleotide polymorphisms.
Yep. So is that a yes, no?
I'd say it's more for population level work.
Okay.
Or even individuals.
Fair enough. Thank you. So this is both of the above, water and soil. And we shall move on. Edna's still there. And for next one, for net zero, it is false. Singapore did not make a pledge to a net zero emissions target. What they made was, go on, Marcus, you know this better than I do.
They did not, they made a pledge according to the Paris Agreement to cut down emissions to half carbon emissions to half by 2030. But they said for the next one was a target. They aim to reduce carbon emissions by 2050, and post 2050 aim to be net zero. So an aim is different from a pledge. So Singapore did not actually pledge to have net zero emission or make a pledge in that sense at all. Okay, so that's our stand. And
you're telling me that it's a technicality?
Yeah.
You're telling me that it's a technicality they got up on? Excellent. And 2, New Zealand is going to leave out methane in their net zero by 2050. So they're going to focus on all the other greenhouse gases, just not methane. So methane will run free and wild like the sheep in the country. And the sheep doesn't need to look so short. Yes, Gretel, same. Same.
Yeah, but in the bill, they did aim to reduce methane to 25 to 49% below current level. So at least they're doing something about the sheep. So I don't know how they're going to do it, either by reducing the sheep or feeding them things that results in less methane being burped out. So interesting to see how they're going to achieve that.
So yes, and then at least they don't want to be rewarded, so that's all right. And the last one is Singapore plans to green up 80% of our buildings' gross floor area by 2030 to meet their pledge to halve carbon emissions. So 80%. So we are going to see a lot more buildings which are towering forests with plants and stuff growing everywhere. And let's go on to apps. Trace Together, the local tracing app, came under scrutiny in the last week regarding Honda that
Nope. I think Kanan
you can get in trouble.
Okay.
And moving on.
Feed.
Alongside 58 other apps. Hello, hello. People, somebody. Am I okay? I think I got cut out there for a while.
Yeah, there was a bit of a freeze. Well, anyway, LeSean's question is, can the answer be none because Singapore didn't pledge for net zero? Yeah, I suppose so. That's a technicality in asking the question. We should have changed it instead of pledge to target in that question. So yes, LeSean, you can get your mark.
Technicalities, nicely done. So previous one was Trace Together. If you guys missed it, because technology hates me. And second one was TikTok, alongside so many other games. WeChat, Clash of Kings, Mobile Legends. Sinway asked why. I think for TikTok, it was more of a content issue. I think a few other stuff, I think it's because of which country they were made in, because they're having a bit of issues now. So yeah, so I think certain apps made by certain countries were banned in India to show their distaste for the country and its workings.
So yeah, relations between India and China have deteriorated because of there was the border clash, I think last year in June. So well, could be a tit for tat kind of thing.
Yeah, so they were like, you can't have any of our money. So yeah. And for the third one is eBird. eBird was the app or online database that was used to report bird observations during the 36th Singapore bird race.
Yeah. So
right.
So let us know if you need us to clarify any answers, because there was a bit of a internet disruption just now.
Yes.
Well, so while that happens, please head over to the trivia sheet to update your scores. So in case you don't have it, it's tinyurl.com/sgstem-trivia. So put in your scores for each round. Evo Lab, we will need you to fill in the beneficiary. Yes. And I think almost all scores, all teams have their points in. And so I'm going to explain the bonus round for those doing it for the first time here. So as you tally up your scores, you notice that some of you are ahead, some of you are a little behind. And so this is the round where you can catch up. Right. So how this works is that you wager a number of points you have in the column there. And what happens is that if you get the bonus question correct, you gain a number of points you wager. So let's say you got all the questions correct in the first 3 rounds. So you get 9 points. If you wager 9 points, you got the bonus question correct, you'll get 9 plus 9 points, 18 points. But if you got the answer wrong and you wagered 9 points, you get 0 points in the end, 9 minus 9. Right, so you lose the number of points if you're wrong. So I see all of you have filled your wager amount already. So Kanan, please review the bonus question. Yeah, everyone's going big.
Yep. Yeah, man. So everyone's home. So you guys got to go big, except for LeSean's team, because they're in the lab. Unless your lab is your home, then yeah. I'm sure it is. Yeah. All right. So bonus round is it comes from the speaker's talk. So in this case, Darren's presentation. What is Darren's favorite group of insects? What is Darren's favorite group of insects? This was mentioned in the talk, so no, we didn't like pull it out of the air.
LeSean says it is our second home, Maya 2019. Wow, when exactly did he say that? You can report him to MOM. All right, so write down your answers or type it in. What is Darren's favorite group of insects? I'm sure it's abundantly clear if you listen during the talk. And so,
shall we? Yeah.
Yeah.
And Darren's favorite group of insects are the Odonata, dragonflies, and damselflies.
Yeah, so that's yay. I'm sure everyone has got it right.
Yeah. So if you guys wagered all your points, you're getting it back now. If you wagered, if you got it right, you're getting it back now. If not, you lost it. And also, I forgot to say, you can only wager what you have. Do not wager a random number, because we know a few sneaky people out there. So yeah, and this is what you can do now. Congratulations for those who won, and thank you for everyone for playing. Please send us a photo or a text of your answers to sgstem.talktrivia@gmail.com, and we will go through the thing and we'll let you know the official winner. Update your scores as well. Marcus will probably be eyeballing it and letting us know the winner shortly. I think we do have a couple more minutes before we need to go. So you can let us know the unofficial winner.
Yeah, so the unofficial winner is Evo Lab, and they're asking, congrats, and LeSean is asking, Darren, please choose what beneficiary you want. Haha. This funding cannot be used to fund your barcode, so
yeah, sorry about that.
Yep, so let us know. Think about it. Okay, so LeSean is thinking. So while they think, I would say, I think the unofficial winner is Evo Lab, 16 points in total.
Nice. Congratulations, Evo Lab.
Second is Sudi the Pangolin, beneficiary WRCF. Third is Fishy, whose beneficiary is Comcrop.
Fishy is second, right? They got 12 and I got 10.
Oh, sorry. Yeah. Fishy is second. Sorry, I can't count. Comcrop beneficiary with grand total of 12 points, and Sudi is third. Sudi the Pangolin, WRCF.
Nice. So congratulations, Evil Labs. Well done. So just send your photo and text of your answers, and then we will probably like send an email along or something to let you guys know of the official winner in case there's been a calculation error, but so far there hasn't been one. So that's good. And while you guys are doing that, let me bring you to our next speaker, who's going to be talking to us 2 weeks from now. Oh, and also there's this thing again. If you guys have not donated yet, or you want to add more because maybe your favorite beneficiary won or your favorite team won, you guys can add more to the pot at PayPal or Pay Now Me.
Okay, so the who was it? Can send the email address again. It's sgstem.talktrivia@gmail.com.
All right, and our next speaker. Do you want to go, Marcus, or do you want me to go?
No, go ahead.
You can go. All right, for next speaker on the 21st of Jan is Dr. Andy Ang from the Raffles Banded Langer Working Group. And Dr. Andy will be sharing with us about the camera traps they have used to study this very rare and very gorgeous primate and all the fun stuff and challenges they face from putting a camera really high up a tree. I am scared of heights, so I hope the rest of you are not will enjoy this talk slightly more than I do. Really hope she doesn't have pictures, from on top of the tree. But I'm looking forward to this talk. The sign up is now live, so you guys can go and sign up. tinyurl.com/sgstem20210121. That's 20210121. You guys can go sign up, and we will see you 2 weeks from now.
Yep, and Andy also says hi. She can't be with us today because we just attended engagement session and she has to eat, she says. Yep, but we see her in 2 weeks' time.
Yeah, so it's a great one. Thank you very much, Darren, for the talk and for staying till the end and playing the quiz.
Thank you. Thanks for having me.
Thank you, Darren. Yay.
Thanks.
Thanks again, Darren.
Thank you. See ya.
Yeah, so LeSean, let us know the beneficiary in the end. Or have you all decided? Oh, you're muted. Have you updated it? Oh, WSCF. All right. So congrats to WSCF. We will tally up the accounts and do the needful. Thanks, everyone.
Awesome. Thank you very much, guys. Have a good Friday and weekend. We will see you soon. And take care.